Hello, my name is Ricardo Rohr, and for my journal article discussion, I chose an article entitled Maternal High Fat Diet and Its Consequences on the Gut Microbiome, a Rat Model. So, for the bottom line up front, we obviously know that the gut microbiome plays a very important role in the health of the mother and the child during pregnancy, as well as after pregnancy, especially since the child does not have a gut microbiome of its own, and it relies on the mother's microbiome to develop the child's microbiome over the course of time. And so, the question that this article seeks to answer is if the effect of pregnancy on the gut microbiome in rats is the same that has already been found in humans. And if if they feed the rat a diet that is high in fat, if this effect will be increased or decreased during pregnancy on the gut microbiome. And so, to do that, this article uses rats as a model organism and various experimental groups that I'll explain in a couple slides later to explore the interactions between diet, reproductive status, and intestinal bacterial microbiota during pregnancy. I will now go over our understanding before this research or the background knowledge. So we know that there are two major bacterophyta that inhabit the human gut microbiome, that being the firmicutes and the Bacteroides. And so these are phyla that are common in the human gut microbiome, and their ratio is often used to measure the overall health of our guts. And so one paper has found that during pregnancy in humans, there has been found to be a decrease in species richness in the gut microbiome, as well as a decrease in the Firmicutes abundances. And then another paper shows that the gut microbiome of obese people have also decreased in species richness and an increase in the ability to extract energy from their diet based off increased levels of Bacteroides. So it is sort of similar. Pregnancy and obesity have similar effects in their gut microbiome. So, why does this study necessarily matter, and what are the broader implications of this type of research? The gut microbiome is closely related to glucose metabolism and digestion in our intestines, and it brings with it the risk of developing gestational diabetes mellitus in the mother during pregnancy. So, this is a common problem found during pregnancy in women, and so this can alter the ability to digest glucose and uptake energy. Um, and then another paper has found that the offspring also can develop health complications if there are health complications during the pregnancy. One of them being immunity problems due to the gut's microbiome not necessarily being very rich or diverse in species. And along with the immunity problems, it can also bring the problem of diabetes or insulin resistance if the gut microbiome isn't sufficiently rich enough when the offspring inherits the microbiome from the mother. And so the broader implications of this is that these health complications could continue to pass down for generations if it is not properly addressed. So, like I mentioned, if the mother has a form of diabetes, then the offspring will be more likely to obtain that diabetes or any other health complications. And if the offspring were to eventually get pregnant down the road, it could pass down to their offspring and it could just continue for generations. So, this is really a problem that needs to be addressed, and it needs to be looked at in the form of looking at the microbiome as one method of combating this and stopping it from being passed down through multiple generations. The author of this paper has two goals, or seeks to answer two questions. The first goal being to establish if a rat model is appropriate for studying the gut microbiome in humans during pregnancy. So essentially, they want to figure out if they can apply their findings from this study or other future studies of using rats to study the gut microbiome can be applied to the gut microbiome in humans, which would be an easier way to look at the composition of the gut microbiome. And so the methods that they use to do this is dividing the rats into two groups, one being standard diet and the other group being a high fat diet. And then within each of these groups, the standard and the high fat diet, they are divided further into a group that is mated and then a group that is not mated. So essentially, there are four experimental groups to look at this goal. And then the other goal that they have is to determine the effect of reproductive status, that being null or unmated, pregnant, and lactating, on the gut microbiome when fed a high fat diet. So to do this, they will expose the null and the mated or re rats to high fat diet over the course of 30 days. And so that will allow them to study the effects of the gut microbiome on pregnant and lactating rats once the pregnant rats have given birth, and then also on the null rats that are just not mated at all. And so for both of these goals, they use the same methods to collect their data. They collect fecal samples at various time increments and then extract DNA from those samples. And then from that DNA, they amplify the 16S ribosomal RNA genes. And then from there, they do Illumina sequencing, which we have learned about in this class. And so we know that this type of sequencing is good for high throughput and high accuracy, especially when trying to figure out the composition of the microbiome. But there are also limitations to this type of sequencing, such as it only looks at short reads, and due to the short reads, it can have a difficult time telling the difference between closely related species, but it's still a very effective and cost-effective method in sampling the composition of the microbiome. So that was used during this experiment. This figure shows the methods that I were talking about in the previous slide. So this here is the first experiment that I mentioned, and these are the rats that are unmated, and this group are fed the standard diet, and this group is fed the high-fat diet, and the measurements were taken at 0, 7, and 14. Those were the days that the fecal samples were collected. And then same for the mated rats. These rats were kept for longer in order to collect data during pregnancy and then also data during lactation. So the fecal samples were collected 0, 8, 21, 26, and day 30. And then for experiment 2, like I mentioned, we have the unmated and mated rats only fed the high-fat diet and also kept for the same duration, the course of 30 days. And then these rats had their samples collected on day 0, 10, 14, 20, 22, 26, and 30. And then same for the mated, and the data was collected during pregnancy and also during lactation. So, for the next figure in the article, the main takeaway is that there was found to be a significant difference in the body weight of the standard diet versus the high-fat diet in the unmated rat group, while there was no significant difference found in the mated rat group. So, to break down these graphs a little further, this first line graph is the unmated rats, and the black dots are from the high-fat diet, and the white dots are from the standard diet. So they found a significant difference between the weights at the end of the 14 days of each of the rats on the diet. There was significant difference in them, and they also found a significant difference from the start of their diet at day 0 versus the end of their diet at day 14. And they also found a significant interaction between time and diet for both of the standard diet and the versus the high-fat diet. Now this bottom graph is the mated rats. And so the black squares are the high-fat diet, while the white squares are the standard diet. And there was no significant difference found between the standard diet versus the high-fat diet. There was a significant difference found from day 0 versus day 32, which is to be expected, but no significant difference in body weight for the diet, as well as no significant interaction between diet and time between the standard and high fat diet. For the next figure, the main takeaway is that there was found to be strong clustering of microbiota based off of reproductive experience in the high fat diet, and not so strong clustering of in the standard diet when looking at microbiota. So, to do this analysis, they did something called principal coordinate analysis, or PCA, where they used variance to look at how closely related the samples were within an experimental group. And then they also want to look at if the experimental groups themselves are distinctive or if they are similar to other experimental groups. So for the principal coordinate analysis of the high-fat diet, we can see that there's distinctive clustering in this group, the unmated rats in this corner over here, and then a cluster of the pregnant rats over here, the squares, and then again another cluster of the lactating rats over here in this corner with the triangles. Now there's a triangle here, but it's mainly clustered over here, and we can see that they're strongly clustered in their distinctive areas, which essentially means that in each reproductive experience, either mated or I should say pregnant, lactating, or unmated, they each have a distinctive microbiota when they are fed a high-fat diet, which sort of explains how they are processing the high-fat foods, and that would ultimately affect their weight gain. Now, for the standard diet, we do see some clustering. We can sort of see that the unmated rats, the circles are mainly in this area, but they sort of blend in with the lactating rats here. There's sort of overlap. And then again, the pregnant rats, the squares here, sort of aren't really clustered. There's really just like a mix of all the data points of the microbiota of the standard rats. And so this sort of means that there's not really a distinctive microbiota when looking at reproductive experience when there's a standard diet. And so the fact that there is a very distinctive microbiota when under a high-fat diet, as opposed to, I guess, sort of random microbiota when looking at standard diet, sort of shows how their microbiota isn't really sort of aimed to extracting the majority of energy from the standard diet. They don't really do that. But in the high-fat diet, they sort of rearrange their microbiota depending on which stage of pregnancy or lactating they're at to sort of digest the high-fat food and extract the most energy from that. And that would affect the weight gain. And so this next figure here is actually from experiment 2 that I mentioned earlier, where they only fed a high-fat diet to both mated and unmated rats to sort of find the interaction between the reproductive status as well as diet, being a high-fat diet, on the gut microbiota and other health aspects within these rats. And so the main takeaway from this figure is that there was consistently significantly more body weight in the mated rats on the high-fat diet than the unmated rats. So here in this graph, we have the squares, which are the mated rats, and the circles, which are the unmated rats. And the measurements of body weight were taken on day 0, 3, 7, 10, 16, 20, 22, 26, and 30. And so they took a lot of measurements, but without fail, on each of these measurements, the mated rats were always significantly had more body weight than the unmated rats. And this fall from here to here is actually when the rats gave birth, which was on day 22. And so irrelevant of whether the rats were pregnant or lactating from day 22 on, the rats still had more body weight, significantly more body weight, than the unmated rats throughout the whole duration of the experiment. The next figure in the article is a PCA of the gut microbiome of the mated and also unmated rats, similar to the PCA done in experiment 1, where they sort of wanted to see if there was clustering or a strong correlation between the mated rats and if it is distinct from the unmated rats of the gut microbiome collected from that. And so they only included data past day 10 of the high-fat diet because they wanted to only look at the gut microbiome after it's gotten used to the high fats of the diet. And so from this PCA, we see that there is clustering of the mated rats, generally over here in these squares, and then in the unmated rats, they're sort of clustered here and then some clustered here. We do see some overlap here, and we see that the overlap is mainly with these darker squares, which is day 30 of the diet. And the article actually points out that when looking at the gut microbiome of humans in the third trimester or later on in their pregnancy, there was an increase in beta diversity of their gut microbiome. But this data sort of shows that later on in the pregnancy of the rats, there was they found no significant difference between the gut microbiome of the mated versus unmated rats. So that was sort of a difference that they found between looking at the gut microbiome in these rats versus the gut microbiome in humans when pregnant, which they took into account when trying to validate if the rat model was appropriate for studying the gut microbiome in pregnant humans. So there is one key difference in there. But we do see some clustering here of the pregnants, like I mentioned before, and here of the unmated. So there is sort of a distinctive gut microbiome, at least earlier on in the diets. The author then wanted to look at the ratio of firmicutes and Bacteroides in the gut microbiome of the mated and unmated rats. As I mentioned previously, firmicutes and Bacteroides are commonly found in the gut microbiome of animals and are commonly used to check the gut health of these animals. And so, when looking at day 0 of the diet of both the mated and unmated rats, we see that the unmated in white and the mated in black, they sort of are around 1 with some variation, of course, but it's close to a 1-to-1 ratio of both firmicutes and Bacteroides, which is ideal in these animals. But once we see multiple days on the diet, like measurements taken near day 5, as well as around day 20 and 30, the ratio of firmicutes to Bacteroides has increased a lot, which essentially means that there are more firmicutes in their gut microbiome than Bacteroides. And this makes sense because firmicutes are often associated with obesity or the high-fat diets that these animals are experiencing. And the high ratio, or just the increased levels of firmicutes in the gut microbiome compared to Bacteroides, shows that there's less diversity in their gut microbiome since there's more firmicutes. Their gut microbiome is mainly composed of firmicutes when comparing to Bacteroides. So in both the unmated and mated rats, there's less gut microbiome diversity, as their gut microbiome is mainly consistent of firmicutes, which is thought to be due to the high-fat diets that they're experiencing. So, in this next figure, the author wanted to look at the relationship between weight gain versus diversity. And to do this, the author uses 2 measures of diversity, one being the Shannon index in the lighter circles, which is a measure of species richness in a sample, and then they use the inverse of the Simpson index in these black circles. And so the Simpson index measures species dominance within a environment, and so doing the inverse of the Simpson index is to see if there's no one species that is dominant within the gut microbiome, but instead there's an even abundance of species throughout the microbiome, which sort of shows that there's a diversity of species. And so in this dot plot, we see that when there's less weight gain, there's more diversity in both Shannon index and the inverse of the Simpson index. And as weight gain increases, there is less diversity, which is found in here. We can see that there's very low Shannon index, as well as very low in the inverse of the Simpson index. And so shows a very nice relationship between less diversity, meaning more weight gain in these rats. And it should be noted that this trend was only plotted in the unmated rats, or the null rats, and there was no figure included about diversity and weight gain in the mated rats. And so I would have liked to see if there was a similar trend in the mated rats in terms of diversity and weight gain, but they did not include that figure in the article. And so, for the conclusion, the author found that rat models of the gut microbiota during pregnancy are, for the most part, consistent with human data of the gut microbiota during pregnancy. The author found that high-fat diets influence the gut microbiota during both pregnant and lactating rats in a similar fashion to human data collected in this paper from 2012. The author found that the reproductive status had a larger effect on the gut microbiome than diet, the standard diet versus high-fat diet, when measuring over a short span of time. But when looking at a larger span of time, like, for example, the span of the whole pregnancy, it was found that the diet had a more stronger effect on the gut microbiota than the reproductive status did. So that was an interesting finding. And then these experiments support the use of rat models and the study of the gut microbiota for maternally obese humans, as they both have similar effects when comparing the data. But obviously, more comparisons would need to be done before this model can be really validated and used in this type of research. Some questions I had when going through this article, one being, what are the potential applications of this information in medicine, such as if a pregnant mother goes to a doctor for a checkup, can the doctor use this information to help guide the mother to maintain a healthy gut microbiome and in turn have a healthy child? The second question I had is, do you believe it is important to maintain a healthy diet throughout the whole pregnancy, or if it's more important during the first half of the pregnancy, or more important during the second half of the pregnancy? I would love to see if there's a difference in that in time span. The third question I had is if a diet in high protein or high in carbohydrates, for example, would have the similar effect of a diet high in fat to the gut microbiome during pregnancy. The fourth question is, do you believe that other parts of your microbiome are affected in similar ways to your gut microbiome during pregnancy, such as the microbes on your skin or maybe vaginal microbes? And then the fifth question I had is, would you use this information to encourage a relative or a friend that is pregnant to follow a certain diet when they're pregnant, or would you wait for another study, maybe conducted on humans instead of conducted on rats? I would be curious to see your answers to these questions. And then these are the references that I used during the presentation. Thank you for your attention, and I hope you learned something.